let's go to this slide, processing from the from retina to visual cortex and beyond. So what happens once the information travels from the retina to the visual cortex? Well, one of the things you have to understand about the way the information is organized in your nervous system is this concept of a receptive field. Okay, a receptive field. A receptive field is an area of receptors that affects the firing rate of one neuron in the circuit, right? So if you've got a cable of neurons that are running to the brain, what you want to do is you want to identify one of those axons inside that nerve and figure out which receptors, say for example in the eye, which area of the retina, which receptors in the retina make that particular axon fire. Okay, and once you do that, you will have identified the receptive field that receptor is responsible for. Okay, and the way we find receptive fields is simply to stimulate a little spot on the eye or on the skin or wherever, stimulate it, and then look and find which nerve is being stimulated by that stimulus. Okay. So what we do is in vision, we just present a very tiny dot to the retina, and then look for the search for the nerve fiber that is responsible for carrying the signal from the retina to the back of the brain. So here's an example, right? If we were to take the optic nerve and slice it like right along through here, right? Just slice it right through here, like that. So it gets cut right about there, right? And then look at it end on end, we would see all these nerve fibers. And basically what we do is we just stick a little tiny glass electrode into one of these nerve fibers and another glass electrode outside the nerve fiber, right? And then record the differential. And just sit there and record and then and then shoot a light, little tiny light at different spots on the retina until this fiber that we're recording from lights up, until it activates. And that's how we know that nerve fiber is responsible for controlling, say, this receptive field right here. Okay. This is a cartoon drawing of a frog eye, right? So we took the frog eye and cut it in half so that we could look at the retina inside the frog eye, right? So there's a receptive field right there, right? So here's the optic nerve. Whoops! Oh God, I always do that, don't I? Here's the optic nerve, right? This whole thing right here, right? This whole thing right here is the optic nerve, right? And then of course running through the optic nerve are these nerve fibers, right? And then here's one right here that we're recording from, this black one. So whenever we stimulate this receptive field with all these little guys on it, with all these little receptors on it, this fiber fires. And so what we're going to say is that receptive field is defined by this spot here, and whenever it's activated, this fiber fires and sends its signals to the frog's brain. Okay. Here's a retina, a frog retina, and here's 3 receptive fields. So what we've done, we've identified 3 receptive fields. So in other words, somewhere along the line here, there's 3 nerve fibers that are originating, that are connected to each one of these receptive fields. Okay, receptive fields are very important. It's a very important concept for you to understand. You have to understand that each the way your body breaks up the area that it's searching for. It breaks up the area that it's searching into receptive fields, right? It happens on your skin, your visual field. This concept holds throughout the body. Okay, so you have to understand receptive fields very well. Okay, this is an example of receptive fields. It's on page 61 of your textbook, right? And what we have here are 2 separate receptive fields, a center surround, and these are what are called center surround receptive fields. In other words, there are some receptors here in the center, and then there's a surrounding area. Okay. Now, whenever the center of this receptive field is lit up, right? Whenever the center of the receptive field is lit up, it sends a signal. So if I were to shine a light right here on this receptive field, just right here in this spot here, oops, it would send a signal out to its nerve fiber, right? It would send a signal out to its nerve fiber. Okay, only if I were to stimulate here. If I stimulate here in the red-orange area, this is inhibitory. It's negative. Okay, what that's going to do is it's going to decrease the signal. It's going to be inhibitory, and so this thing is this receptive field is not going to fire. Okay, this receptive field is the opposite. If I shine a light here, it'll be inhibitory, so the receptive field will not fire. But if I shine a light out here, it will be excitatory. It'll stimulate firing, and it'll then send a signal. Okay. Now you can imagine that you could actually have a whole bunch of these stacked next to each other, right? Just like that, right? You could have a whole bunch of them all kind of like overlapping each other, just like that. With a bunch of positive receptive fields, right? If you think about it, that would be a really good way to perceive a line, right? We think this is how we perceive lines and edges, right? Is these receptive fields all get lit up, and the ones that are along the line are getting stimulated, and so they work together to fire. Right? And we can also perceive edges with this one as well, right? If there's an edge here, if this is not getting lit up, right, this would be an edge. Okay, so it's kind of a neat way. You have to imagine that there's millions and millions of these receptive fields in your retina. Each one of these little circles is made up of a few dozen rods or cones. Okay, so these receptive fields are just made up of a few dozen out of millions and millions and millions of receptors. So there's millions of these receptive fields that are just laid out in a map on your retina, and they get stimulated based on what light is hitting them. Okay. So the output of the center surround receptive fields changes depending on which area is stimulated, just like I just showed you. The highest response only when the excitatory area is stimulated. So in the one that has the positive center, if the center gets stimulated, you get the highest response. The lowest rate is only when the inhibitory parts are stimulated. So again, if you go back to the one with the high center and inhibitory surround, if I shine a light or touch the area, the inhibitory area, it won't fire, right? It'll be inhibited, right? What if we stimulate both, right? What if we go back here and I shine a light that hit, whoops, oh. I wish that pen would stay on. What if I shine a light that hits both, right? So it hits the positive and negative. You get an in-between response, right? Now, this relationship between the center and the surround on these little donut receptor fields affects how each receptive field is going to fire, right? So you get the highest response when the excitatory area is stimulated. So in this guy here, if the center is stimulated, or in this guy here, if the edge is stimulated, it shoots, this receptive field sends the highest output, right? If it's stimulated in the inhibitory area here, or in the middle here, it sends the lowest response. Okay. Now you have to ask yourself, what happens if it's stimulated in both? What if what if a stimulus hits, say, right here? Right. Well, it's going to be an intermediate response. It'll be not too high, not too low, and it'll vary depending on how much of the negative it got and how much of the positive it got. Okay, so if you think about it, and you have to remember, these receptive fields. There's millions of these receptive fields, millions of them that are just arranged on the retina, right? And so they work together to collect all of the edges and the shapes, and to define all of the edges and the contours and the shapes of things that you see in the environment. So here's an example, right? So this is a a receptive field with an excitatory center, right? So if it gets stimulated on the center, it fires, and if it gets stimulated negatively, it doesn't fire. So it's getting a little bitty stimulus here in the middle on its excitatory parts, and it kind of fires, right? It's doing okay. But then when we widen the stimulus to light up the entire area that is excitatory, it fires a lot more. If we expand that more and go from the inside of the excitatory area and start getting some of the outside inhibitory area, it slows down. And then when we light up the whole circle, the whole receptive field, it slows down pretty good, right? So if you think about it, this is a really neat way to define how big or how wide an edge is, right? Because remember what you're going to do. You know, your retina is full of millions and millions of these things, right? And they're all stacked next to each other, right? They're all stacked next to each other, right? So you've got these excitatory circles all stacked next to each other, and it turns out that that's a really good way, for example, to make a line, an edge, right? If they all get lit up in the same way, they'll work together to form a line or an edge, which is kind of cool. Here's an example of the receptive fields wired up to the nerve fiber that we're recording, right? So what we're doing is we are recording from this nerve fiber right here, right? So our electrode is actually stuck right here in this nerve fiber. So what happens when we only light up the center, right? So this is an excitatory center, right? And what you're going to see here is that these three guys, these three receptors, 3, 4, and 5, are all directly connected to the nerve fiber, to the body of the cell that generates the nerve fiber. Okay. So when only the middle one is stimulated, this guy here, right, it only gets stimulated by one, and so it fires a little bit. But if it gets stimulated by all three, it's going to get positive input from all three of these guys, and it's going to fire like crazy. Right? What happens when one of these guys gets stimulated? Well, if one of these guys gets stimulated, they of course fire, right? But they're connected to this interneuron here, this one of these amacrine cells, and what's going to happen is this guy is a negative. It sends a negative inhibitory signal, right? So these guys are going to stimulate this guy, but this guy is going to inhibit the nerve fiber. Okay, so that's how the wiring of the retina creates a receptive field that has, say, for example, a positive center and an excitatory center and an inhibitory surround. Okay, same over here, right? If this is if anywhere outside here is stimulated, the receptors that are sitting there watching that area are going to stimulate. They're going to fire. But they are connected to an inhibitory cell. And that inhibitory cell, in this example, the inhibitory cells are A and B, and E and A and C, right? If either one of those gets stimulated, they send a negative inhibitory signal to B and say, don't fire, right? And you can see how if we have a stimulus that goes on both sides, on the positive side and on the negative side, you can see how that would result in a kind of a medium firing situation, right? Because in this example here, 5 would be firing away and stimulating B, so B would be firing away. But 6 is being stimulated, and it's telling C to inhibit, so it's blocking the effects of 5, right? So that's why you get the kind of the middle firing if you have the stimulus crossing both the negative part and the positive part. Hubel und Wiesel. Hubel and Wiesel. They are the guys who won a Nobel Prize actually for discovering these receptive fields in the visual system, right? What they did was they said, okay, what they did was they mapped out the signal as it goes from the retina all the way back into the brain, into the cortex of the brain. And they looked at these pathways in cats. Okay, so they found the signal, and they found out that the signal travels from the retina, and it first stops at the lateral geniculate nucleus in the thalamus. The lateral geniculate nucleus in the thalamus is the thalamus itself is responsible for partially integrating and collecting all of the sensory information from our body, not just vision, but also hearing, smell, taste, touch. It all gets sorted and integrated in the lateral geniculate nucleus, and then it makes its way further up into our cortex. The first stop for visual information is the primary visual cortex. It's in the back of your head in the occipital lobe. And then from there are pathways that take the analysis from that area and take it to the temporal lobe, which is here on each side of your head, and the parietal lobe, which is on the sides and the top. And then finally, whatever analysis comes out of there comes to the frontal lobe here. The frontal lobe in humans is where we do most of our higher level thinking and planning. Okay, so it's very important that anything you see makes it there so that you can deal with